Imagine a world where we cannot follow the course of a disease by PCR tests. Imagine a world where we don't have the ability to develop a vaccine against a new disease in a matter of less than a year. Imagine a world where the development of a new treatment is not based on scientific understanding, but it's based on trial, error, and witchcraft. This is the world without biotechnology. This is the world of the 14th century, where the bubonic plague wiped out over a third of Europe's populations. This is the world of 1980 to 1920, where the flu pandemic killed over 100 million people. My name is Ehmke Pohl. I'm a professor for structural biology at Durham University. And today I will take you on a journey from the depth of the Atlantic Ocean to the hot geysers in Iceland to our research facility here in Durham. In the last 18 months, we have not only seen the first pandemic in over 100 years, but we have also witnessed an unprecedented collaborative effort of researchers worldwide to combat the disease. Biotechnology is based on the use and optimization of natural proteins or enzymes that are capable of highly specific molecular tasks. For example, the cut and paste of DNA, the amplification of DNA for genetic testing, the production and stabilization of therapeutic proteins for COVID-19. Many of these protein tools in biotechnology are originally discovered from viruses. However, not the viruses that infect humans, but the more benign kind of viruses that infect bacteria. In fact, viruses are incredibly diverse. Viruses have evolved to infect almost every organism on the planet. And there are more viruses on the planet than there are stars in the universe. However, the vast majority of these viruses have not been discovered because viruses are notoriously difficult to study. Since viruses replicate and mutate so quickly, their genetic material is believed to be the most unique and the most diverse on the planet. Hence, this has often been called the dark matter of life. With the fast growth in biotechnology, there has been an increasing demand for ever more sophisticated tools in biotechnology. So in early 2016, the Virus X team, consisting of about a dozen European research groups, including our own here in Durham, and 3 small biotech companies, started their project supported by the European Union. Our goal was to find the most unusual and novel viral proteins from extreme environments. The most otherworldly of these locations is probably Loki's castle. Loki's castle, named after the Norse god of mischief and made famous by the Hollywood franchise of Marvel movies, is located more than 2,500 meters below the surface of the North Atlantic Ocean. Here, hydrothermal vents spew hot water almost 300 degrees centigrade into its environment, creating a unique habitat of plants, bacteria, and viruses. There's only a handful of submarines that can explore those depths of the Atlantic Ocean. Our partner, Professor Ida Steen from the University of Bergen in Norway, has led expeditions where she controlled a submarine remotely operated, where she collected samples from Loki's castle for analysis. The organisms itself would not survive the trip back to the surface. However, their DNA would. In fact, a single sample contains the DNA of hundreds, if not thousands, of microorganisms and their viruses. In the next step, the proteins encoded by the most promising genes can be produced, purified, and analyzed in our laboratories here at Durham University. Over the last 5 years, the Virus X team has explored many other extreme environments, including hot springs in Japan and the famous geysers in Iceland. Over the past 5 years, the Virus X team has explored a range of other extreme environments, ranging from hot springs in Japan to the famous geysers in Iceland. And in summer 2017, I had the privilege to join one of those bioprospecting expeditions into Iceland. Watching the solstice sun not setting in the Icelandic summer and collecting tens of liters of hot acid was a truly remarkable experience. During the course of the project, the Virus X team has been able to identify over 50 million new genes that are now being curated in the Virus X gene database. In addition, we have been able to produce and analyze several hundred of new proteins in great detail. As we all know, in January 2020, the world irreversibly changed. And in March 2020, many scientists worldwide realized what was about to happen. During the first lockdown, we immediately formed a COVID-19 task force with the goal to pool our resources and our field-specific knowledge to combat the virus. Since we are basic scientists with no expertise in vaccine development, it was immediately clear to us that our contribution would be in the test and trace regime. A number of countries in Asia and the Pacific had already shown that a working test, trace, and isolate system was paramount to contain the pandemic. At that time, 2 different technologies were quickly adapted to COVID-19. PCR tests that remain the gold standard today require expensive equipment and infrastructure. Lateral flow tests, on the other hand, are easy to apply, but they are notoriously unreliable, in particular when the sample is taken by the subject and not by a healthcare professional. So we decided to look at a third technique called LAMP assays. LAMP assays are almost as reliable as PCR tests, but they only require a water bath that needs to be heated at 65 degrees Celsius. So they can be used at the point of care just like a lateral flow test. In addition, LAMP assays offer the potential to detect and distinguish between different virus mutants. Of course, a number of groups worldwide started to work on LAMP assays, and we were lucky to share our experience with other groups. However, we had 2 trump cards. First of all, we had our gene database with millions of new undiscovered genes. Secondly, we had already established the workflow to produce and test proteins at a larger scale. So when the laboratory slowly opened up last summer, we identified auxiliary proteins in our database that have the potential to improve LAMP assays. Together with our partners from the Norwegian biotech company ArcticZyme, in particular Dr. Ketil Strøberne, we were able to show that our auxiliary proteins significantly improve LAMP assays. Importantly, while focusing on COVID-19 applications at the moment, there's a huge potential that these auxiliary proteins and many other proteins find applications in modern biotechnology. I hope this example shows how a project that started as a purely basic research project can have real impact. So the next time you're submitting a sample for testing, you may unknowingly encounter a component that comes from the depths of the ocean. Thank you very much for your interest, and if you want to find out more about the Virus X project, please visit our webpage.